Today I'm going to be helping you prepare for the upcoming lab practical. This is review session 3 for those of you who take my section at QC. I am now going to just quickly get into things and start reviewing some stuff. So I'm going to switch applications and ask you questions about the content. I'll draw a little bit on images. I'll give you a few cues and guide you the best I can. We're going to be doing the last couple of labs for the most part. So those of you who are here for the first time, don't assume that this will be the only content because this is part 30 for my class. So now I'm going to share an application called OneNote. And I'm going to throw a lot of animals at you. I'm going to let you look at them for a minute. You should be asking yourself, can you name these? And how do they relate to each other? In other words, if these were cards on a table, how would you cluster them to show evolutionary interrelationships? Your sound is still working, by the way. I haven't been talking. Right now, I'm just working with what's been dumped out all over the floor and sorting it based on phylogenetic affinity. And you have to start somewhere. So this is rudimentary sorting that I'm revising as I go. Obviously, I'm trying to get this away from this. And I don't see any of its kin nearby. So I better get it out of the way. I found something here, and I'd like to keep these together. I'm clearly going to have some sorting to do in here, but first for the major groups. I know where this goes. Here. More specifically, here. So I want these together and this one close. This is a tapeworm, but I don't want it near this worm. This one's easy. It has some moving up to do, though. I'm going to start to shrink things down. Because we're getting a clearer picture. I want these on top of each other because they're the same. Just different views. I want these very close. Now this. I want close to these. But I want this to stay closer to this. This so far has nobody who we've seen that has a close affinity, such as phylum is what I'm considering to be close right now, which means that I have at least found a friend for the tapeworm. This. This I'm moving somewhat. I'd like it to have some space from these three. I want these three close, very close. I want this closer, and I want this nearby. This has nobody. The same is true for this. The same is true for this. The same is true for this still. Nobody near. But these have affinity. These have affinity. These have affinity. These have affinity. And these are rather alone. So you should ask yourself, what is the name uniting these? And I think we'll go back to this. But now we'll get more specific. Let's look at sponge-like things. This we just saw is a glass sponge. And we actually would have handled a few of them in lab. Were we in lab? So this is a glass sponge. And this is what they look like close up. This is a dried specimen. They're very delicate, and they have a shared derived characteristic shared by this, which is a different kind of sponge with a totally different body plan. We're going to look at this later because, spoiler alert, this is not a sponge. I named this section sponge-like creatures to show you some things that could be mistaken for sponges and that are sponges. This is in fact a sponge. So these all belong to a phylum together. Did I spell this wrong? No. Yeah, it's a hexantinellid. Okay. This So I definitely want this lowercase l. Same with this h, because this isn't the name of the taxon, but that is okay. It's descriptive. So this is a glass sponge. This is a different kind of sponge with a leuconoid body plan, which is more complex than than an asconoid body plan. So there are 3 sponge body plans. And in the original page that I showed you, we saw this, Grantia. So Grantia is a genus, and that's things that you may need to say for the practical if you're asked for the genus here. Grantia, that's an A, not a G. So this is a cross section, be taken just like this, from a Grantia specimen. And you can see that this is not a complicated way for water to get in to cells. So it's going to be up to get into the cavity inside where there is filtration and nutrient absorption and oxygen absorption. And so there are a couple of key terms I want to write for you that you yourself can make the association with. I won't cheat you out of that. It's actually a much, much better exercise. I'm trying to close this. In any case, This one is asconoid. It's also called grantia. So some other sponge vocabulary words. Porifera. That is the phylum. Also, choanoflagellates and choanocytes, which I'm not going to get into because it's specific anatomy. But these are some vocabulary words that are good to know. So this is a leuconoid body type, and we just saw an asconoid in the. Okay, so some other sponge-like creatures. How can you tell it's not a sponge? Well, for one, this tunicate, which is far more complex than the sponge, has two openings. It also lacks the openings. This is pretty solid and leathery. Then there's this, which if you're very new to the material, it could look spongy with a lot of imagination, but it has a mouth, an anus, these tentacles. It cavity in water goes in, water goes out, water goes in, water goes out, and that's all. This is far more complex than that, and this also contains a complex body plan, as does this. It sits there, it's marine, it's round, it looks pretty simple on the outside, but it has a digestive system. It also has spines. This is a view of the tunicate plan. So you can see that there's an in-current siphon and out-current siphon, and this is just to show you that there are some things that look similar, but you'll have to make me know that they may be sponge-like or round or have some superficial similarity, and you have to learn how to tell them apart. So this is an echinoderm. This is also an echinoderm. These are poriferans, sponges. And these are urochordates. So let's move on to clam, which is a mollusk. This is a clam. This is, it's not even a mollusk, which is why I'm moving it away. But I have it on this page for a reason. So you may have pointers to, such as this, and you'll be asked to identify one or more parts of the anatomy. And at the same station, you be asked a question about its taxonomy or function of one of the organs. But you may have to side the clam, meaning point out which is the right side and which is the left side. And once you learn that, that's pretty helpful because you can count on the anterior adductor muscle being on the what to us is the right side. So think of the clam as anterior and posterior, And the umbo, which is the bulge at the hinge of the clam, it's pointed a little bit in most bivalves, not all of them, but in this clam, which I'm pretty sure is a quahog, it leans for us, so that's nice. So whichever way it is leaning, it leans away from the posterior and toward the anterior. So then you'll have these two large muscles that are used to keep the shell tight together. And now you can identify this one as anterior and this one as posterior. The posterior one is usually a little bigger, by the way. Another major feature, which is it's usually pretty hard to pick out, and I'm going to delete some of these lines for you, is the gill. There's a large gill embedded in the body, and it's thin. It has linear segmentation, and the gill tapers a little bit toward the posterior. So the larger side, the larger end of it will be on the anterior, and the slighter side will be posterior. So that's one way to keep track of the gill, but the best way is to be able to recognize the tissue. And to do that, you have to differentiate between gill tissue, which is in red, and the tissue below the 5 arrow that I'm moving the cursor along, which is the mantle. So if you can differentiate mantle tissue from gill tissue, then that's very helpful. So try to be able to do that, and then know that and then the foot, this meaty part, and this is like a foot. I think it's actually a proboscis, but the foot is for pushing the clam along a little bit, and it points forward a little bit. It leans forward. It's like a tongue, and like the tongue is wagging toward the front of the body. And so I'm not going to tell you which is which, but you should know which is the in-current siphon and which is the out-current siphon. And they're stacked one on top of the other. So that's some homework to do, in-current and out-current. Also, heart and stomach. So if you know, I didn't want to put everything up here at once, but learn those things and that'll be good. So this is a mollusk. Mollusca. Bivalvia, which includes a number of shelled organisms. But then there is this. It has 2 shells. It looks rather clam-like, but it is absolutely not a clam. It's a member of Brachiopoda. And its anterior is here, and its posterior is here. And this is its lateral part. So it's as if it is standing with the toe here and the head here. And with this animal, it's as if it's on its side with its toes here and its nose here. So that's a bit about that. So learn those. Also, what is missing? From mollusca, there are other organisms that could be here if this was not just a clam and brachiopod slide. There are a number of taxa that separate these. but closer to the clam. I'll zoom in a little bit. Those come to mind. Gastropoda and cephalopoda, which are the snails and squids, octopuses, and all of those. So an octopus is more closely related to this clam than to this brachiopod. So that is an important lesson. So I showed you sponge-like. Now these are worm-like animals. A few of them are true worms, and which means that they're annelids. So I'm going to give you a minute to look at these and take them in. And determine which are true worms and which are simply and this will throw a wrench in your gears. Oops. Oh boy, sorry. Okay. So this page contains a number of worm-like creatures, only some of which are worms. Let's look at this a little closer. Oh, this gives it away a little bit. Polychaete worm. But is a polychaete worm a true worm? Is it a member of Annelida? And it has something called a lophophore, which should place it in your mind in the lab where we covered blank. And that word is so now you can also be asking yourself, for each of these, is this an annelid that I'm looking at? Is it a lophotrochozoan? Is it both? And now I will start feeding you words in the event of multiple choice. So I'm giving you a word bank, basically. Genus. I'll zoom in on this a little bit. All of these terms are attributable to animals on this page. And these are taxonomic. Oh, probably spelled that wrong. Sorry. Damn. All right. Acorn worm. I'm going to zoom out again. And I'm going to start moving some animals around. This, zoom. Way away. Not too far from these, though. Let me add ecdysozoa. And another one. I'm saying it because I know you can't see it. Arthropoda. Ooh, and I forgot one. Nereis and Polychaeta. So I'd like to see, keep these close. Uh-oh, I have another one. Oligo, oops. I have Oligochaeta and Polychaeta now. And just one more. Clitellata. Okay, so I'll break this down. Really would like to go away. Oh, there we go. Sometimes you get what you ask for. So these are true worms over here. They're annelids. And this is Lumbricus terrestris, the earthworm, for which you have a major dissection. This is Nereis. It's a polychaete worm. And this is Hierudinia, the leech. And you could see a bowl with preserved leech specimens in it. So these are all annelids. These are actually very close, even though this one, the Phorinid, which, oh no, one more. Phorinid. So I'm going to draw some associations where there are associations. Now that I've organized them. Hierudinia, Lumbricus terrestris, Lophotrochozoa for all, Nereis, Oligochaeta, Polychaeta, and this. Again, another color. Clitellata for both. So this looks messy now, but oh, and Phorinid. But that's just how the words are stacked, rearranged. They could come across nicely. So I realize that this is a lot to memorize, but you can. The best tool is really, you can't beat reading the lab manual, and to read it a couple of times and keep reading it. I think one of your quiz questions at one point was, what do leeches and earthworms have in common? They're members of Clitellata, for one. And I want to do one thing right here, so I think it's important, because these are radically different. This velvet worm is an ecdysozoan. This is an annelid. and Alophotorozoan. So they both are kind of similar in appearance. So if you saw two specimens floating in bowls, look at these little slinky arms. They don't have any kind of little spikes or hairs or bristles. And these, the polychaete worms, the genus Benares, has bushy bristles on the ends of the legs, which introduces an anatomy term, which is really important. Setae are shared derived characteristics for annelids, and they are totally absent on this guy, which is Onychophoran, which is a velvet worm. And it has a much more complex body plan. So ecdysozoan is what these are as well. So that's something that these have in common. These two stand alone in that they're myriapods. Myriad of legs. And above that, and ecdysozoan is above Arthropoda. So these have jointed legs. So you should learn centipede v millipede on your own. I've covered that with my class really well, I know. So, oh, and then that, I said I was throwing a wrench in your gear. And by that I mean I'm introducing a hemichordate, something just multitudes times closer to people than any of the previous. And this is an acorn worm. And I'm sure if you're asked to identify it on some level, you have acorn worm if nothing else. So distinct from an earthworm, easily, which also has a band like this, which in an earthworm is called the clitellum, or in a leech is called the clitellum. You don't always see a clitellum in a leech, but I believe they are there, or they were lost at some point. But this is a proboscis, and there's an obvious seam in between the proboscis and this collar, which can slide. Another way to tell is that there's no opening here because the mouth is down here. In a worm, the opening would be here. Also, gill pores are present on the on this hemichordate. Which is Enteronusta. Those are the acorn worms. And above that, Hemichordata. Also, there's something missing. If this were a true worm, it would have segments, lots of body segments, and it doesn't. Worms and leeches do. Okay, so I'm going to move out of this and talk about the earthworm specifically, but not too much because I've gone over this a lot with my class. So this is a moment from the PBS dissection video, and this is a worm that I drew that is stripped of some of its organs to emphasize the digestive tract. So these are terms to know for the worm, and you should be able to order them. And if you can start just by pharynx, crop, and gizzard, pharynx is always going to be first in anything because that's the mouth opening. It's first. Crop is where there's some digestion, and it's also a holding space before food can get to the gizzard, which is grinding, and it's very hard and compact. And then intestine follows. So if you can just at least do these three, then that's really good. Remember that the worms have setae on them. So I'm sort of stepping aside and drawing attention to another feature entirely, nothing to do with the digestive system. But if you rub your fingers against a worm, it's not that slimy unless it's wet. And even if it's wet, I thought if I hit E, then that would be a hotkey for erase, but apparently not. Okay. So you should keep in mind that this is the bare digestive tract that I'm showing you. I've stripped away a few things. So you should, if you can superimpose what those missing things are and where they go onto the tract, then that is good. And of course it has a well-developed gut because it is dot, 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 dot. What am I getting at? Hint. This is a hint. And the answer is one word. Completely separate from that. Is it a protostome? These are good things to know. And you can ask yourself that about these as well. Which of these are protostomes and which are deuterostomes? It's very easy. Only the hemichordates here are deuterostomes. Everything else here is a protostome. So I've alluded to what is missing. And this is a very freshly open worm. So these are all reproductive organs. They could be male or female. I'm not entirely sure. And these bits here are what remains of aortic arches. So a collar of rings that is heart. And I actually did an illustration for that too, but I didn't include it here. So no, the circulatory system over the worm's digestive system, and its reproductive organs, and perhaps the role of the clitellum in reproduction, which, because these worms are cut open, you cannot see. So I showed this to my class last Friday, and I scribble over this Dugasia slide. It's a flatworm to show that there's a digestive system layer, there is a nervous system layer, and there's an excretion layer. And if you can differentiate, then that is good. It helps to be aware of the layering. And if you look at a schematic, an illustration of a flatworm, which is a platyhelminth, platyhelminth is broad. That's a very, very, very high taxon. Then you are in good shape. And I'll go ahead and erase all of this now. So you could get a slide such as this, where you will have to identify one of these things. Probably not trunks or anything like that, but you should know that this whole thing is gastrovascular cavity. This is the pharynx, which can be distended, and that the mouth is at the end of the pharynx. The auricles, we didn't talk about that much with my group. They're simply the triangle. I'm hearing a lot of commotion outside. Anyway, okay. So, okay. The auricles and eye spots. And here are some protonephridia, is part of the excretory system. The ganglia would have been nervous related, which I just erased, and the pharynx is still shown here. So this is the type of platyhelminth, which is high taxonomy. Below that, I'm going to shut my window. All right, Washington Heights. Still not quiet, even during a pandemic. Okay, so I'm going to talk about the frog for a little while, which should be pretty fresh because it was yesterday. And I'll let you have a look before I speak. And while you are looking, I'm going to draw some margins for you. This has one name. So I've showed you the lungs, and they are pulled up a little bit. The lungs are in purple. And lung tissue is always very folded and convoluted and soft looking. And the heart is up here, and it's got 3 chambers. These, of course, are liver lobes, and somewhere underneath it, there will be a gallbladder. In red, that's the pancreas, which is always going to be close to the stomach. And this is a long, long, long digestive tract. And this yellow mess, those are fat bodies. It's just a different type of fat storage than we are used to seeing. And there is more to the frog. Kidneys toward the back. My class, we spent a while on this yesterday. So I'm going to go to things that we have not looked at in a while. particularly this because it includes sea urchins. I spent quite a while on starfish arms. We barely made it to sea urchins. These are all echinoderms, and this is a sea cucumber. And it may serve you to know what these organs are. Not only that, what's the name of its group? Holothuridians or Holothuridia. So know that not shown are tube feet. If you open it up, you can see the system for the tube feet on the inside. So what are these organs? Another question. Where is the madreporite? So that's something to always keep track of when you're looking at echinoderms. In this case, the madreporite is on the inside of the body. Then there is the sea urchin, Echinoidea. I just wrote Holothuridia. Echinoidea. And this is mostly gamete. This, a brittle star, lacks a number of properties that that the starfish has. So no ambulacral grooves, among others. Since this is a review, I won't be going into all the details of everything. You should know, you may need to know, is this the mouth or the anus? What is the name of the surface? Anatomically? The surface? I'll throw out aboral. So there will definitely be something on the anatomy of a starfish arm. And it could be a cross section like this. You could, I think it's very, very likely that you'll get a dissection picture. So try and keep track of where the gonads are. There are only 3 systems you have to worry about in here, 3 layers. One is digestive, one is for motility, and the others are gonads. And the gonads are on the bottom, the ventral surface. They're low, they're down here. The ampulla system, the one that keep the pedicularia going, those are the ends of the tube feet. There's that system that's situated mostly in the ambulacral groove, it's hydrostatic, and hepatic cecum, which is all digestive. So remember those. And here's just another showing of the same thing. It's a very nice looking starfish dissection. Okay, so now I'm going to talk a little about Amphioxus, because you may have to identify a piece of its anatomy. So a diagram like this would help you. But as I've told my class many times, notochord v. dorsal nerve cord. And I pointed out once that here, I believe that this broken line was dorsal nerve cord, and notochord is above it. So which is the notochord and which is the dorsal nerve cord? And the dorsal nerve cord is higher. You probably will have to differentiate that at one point. Even down here, it's hard to control my men a little bit. So it's higher up, and it is not for structure. Okay, so the blue is the nerve cord, and the yellow is notochord. There's a list of 5 chordate characteristics you should know. We did echinoderm. Perch is very fresh, so I'm going to skip that. But squid may benefit you to also to know the surfaces of the squid. So dorsal for the squid is up top at the end of the fin. Ventral is the surface where the mouth is. Anterior is the opposite of this. This is the posterior, and this is where the siphon is. So the siphon is on the posterior. Female squid. So with the squid, you may be asked to identify the branchial hearts or the cardiac hearts. And know which set does which. The ink sac is here in gray. I highlighted it a little bit, and here's what it would look like on the outside. If you hang on one moment, I have a few squid pictures that I would like to grab. Actually, I have a squid dissection video that, how can I make that? I'm going to put it on my website. I'm going to put my squid dissection video there. It's a 10-minute video. Okay, so those are some recents. I think we're going to need to have one more review next week because it really didn't cover arthropods and ecdysozoans very much on this round. There will also be some other reviews by others instructors, and you'll be kept up to date on when those will be. In fact, let me give you some times now. But you will need the links. Okay, there will be, I'm going to type this into the messages. And I'm going to stop recording.